Good afternoon, everybody. This is JC on a bike. 73 degrees in Pittsburgh, Pennsylvania today. I'm trying to take advantage of the last 70-degree day in Pittsburgh and do a little journal club on the way home. Trouble is that this journal club that's been selected for me by the Undergraduate Journal Club is extremely cursory paper about a discovery in the parietal cortex, and I'm not really sure what the objective is of this paper in terms of what we want to get out of it and where we want to go from it. So I'm just thinking about parietal cortex here as I talk about it, and the idea is that this particular region of parietal cortex in a primate is doing something with the visual field and with retinal space. This is described in figure 1 that shows a sort of cartoon version of what's happening during the task. And essentially what it is that there's a fixation point, and outside of that fixation point, the fixation point being where the gaze of the animal is fixed. Outside of that fixation point is this dotted circle area, and in that dotted circle area is the visual field that this neuron responds to. And when there is a object in that visual field, an object being the star in this case, then the cell will respond with spiking. Remember where this Amazon place is, but it's not down here, I see. Sorry about that detour. So what we're talking about again in parietal cortex is, what does parietal cortex do? And I think this has been a big question for a long time. People still don't know. One of the things that the parietal cortex is most famous for are mirror cells. Mirror cells are cells that seem to fire while an animal is watching some other animal do something that it knows how to do. Like cells that would fire when you put on your coat because I know how to put on my coat. This man is braking and going in front of me. So the parietal cortex is a pretty enigmatic higher order cortical area, processing visual information and also body position information, and there's a lot of things that the parietal cortex does. We're not really sure what all of it is. And in this paper they're looking at a particular nucleus of the parietal cortex, which may or may not, the same nucleus may or may not exist in a rat. In a rat we may only have parietal cortex and nothing else, but in the primate brain we break the parietal cortex up in a few more places, and this lab has recorded from a particular area in the parietal cortex that seems to show some form of visual information being queued up ahead of time before the eye executes a saccade. And what this is purported to be evidence for is the idea that the parietal cortex has a retinotopic map, and it can more or less make predictions about where the retinotopic map is going to move and how it's going to move, and then indicate expectations for a stimulus presence, and even indicate a stimulus that was there but not there anymore. So in this paper, they just basically show this one phenomenon and leave it up to you to decide with really two data figures whether you believe their interpretation of the data. And all they're going to do is show you spiking. And so in the first figure, it's just a cartoon, right? And what they want you to see is that during a saccade, there are three sort of main conditions. The first one being the starting condition, where you have a gaze in the middle, and then some receptive field that in this picture is in the cloud. And then if you move the gaze of the animal, then where that receptive field is relative to the image of the tree and the mountain and whatever else, then it will be different. And so what they seem to show in this paper is that the parietal cortex remaps to the new gaze position to indicate the expected content of the new visual field before the saccade is executed. I think I said that pretty well. But let's just say that in each of these figures, then, what they're trying to do is show you the spiking in a parietal cortex cell relative to the appearance or disappearance of the asterisk stimulus in the receptive field indicated by the dotted line circle. And so the eyes are fixed on the dot or the small square, I can't remember which one it is, and then they move from the first one to the second one following the arrow. And in the figure 2 and figure 3, you can see the horizontal and vertical movement of the eye so that you can know that the animal is not looking directly at the stimulus, the asterisk stimulus, but he's maintaining his fixation on the gaze locations where he's supposed to. He's following the task. And so you can see the horizontal saccade, but there's no vertical. And so then what they do is they just show you the different Stop, lady! So then they just show you the different conditions, basically. First condition is when they turn the stimulus on, but there's no saccade. And so they turn the stimulus on. A few milliseconds later, the neuron starts firing in every trial. The next one, they show two examples. This is figure 2, where they show a saccade where the stimulus comes into the field as a result of the saccade, opposite of one. And then they show two takes on this. One is a stimulus onset centered analysis, and one is a saccade centered analysis. So what you see here is that saccade is not activating it. And that's the main point they're trying to make here, is it's not the saccade, it's not the eye movement, but it's the presence of the stimulus in the receptive field that gets you the firing. And so obviously if they make a saccade where there's no object in in the receptive field of that cell, then there's also no firing. Now this is still just correlative data, right? They're still just showing you that it appears that this cell behaves this way. And so one of the things when you read a paper like this is you have to read through the text and read through the methodology and read through the results and find out how many cells they recorded from in these 2 monkeys and how many cells show the responses that they're advertising. Because it is not all of the cells. If it was, it'd be a pretty extraordinary paper, right? So be sure you figure that out. Another very interesting question to think about is where are they recording? Are these superficial layers? Are they deep layers? We know from other cortical regions that vastly different firing patterns and encoding regimes can be separated by simple layers in the entorhinal cortex, for example. So it wouldn't be unusual for this phenomenon, say, to only be in one layer and not to be in all layers. So it'll be interesting to see if they know anything about where they actually recorded from. I guess they don't. It says that this Michael E. Goldberg is at Columbia University now, so I'm not really sure who you're going to talk to for this paper, who you're going to be asking about it. Here we go, super genius. Wow, that is some junkie style vehicular operation.
Hey, nice scooter, man. Get down on the ground. You must be crazy. Bulletproof bicycle.
Hey, thanks for joining me on my ride. That was an older paper regarding the parietal cortex. The parietal cortex is still kind of an enigmatic cortex. We've done a lot of work since 1992. So this paper shows one aspect of one particular region of the parietal cortex, but I think it's important to emphasize that we've learned a lot about other cortical regions that show that cortical cells encode very specific things on a layer-to-layer basis. And if the parietal cortex is anything like the entorhinal cortex or the visual cortex or the prefrontal cortex, then in each layer a different operation is going to be happening, and therefore it's important for them to know where they're recording these cells, what proportion of the cells they record from are showing the advertised output, and then to scrutinize better sort of the way that they do the experiment to be sure that you believe it. And then I think, of course, you know, since 1992, a lot of papers have come out. So hopefully in the discussion of this paper, some newer data will be presented to let you know how this particular region in parietal cortex has evolved, our understanding of it has evolved. Thanks for watching. If you liked it, subscribe, and I'll see you next week on the bike, Journal Club. See you.